Hello there, my name is John Baugh and I'm the Programme Director for the MSc in Experimental Physiology here in the UC School of Medicine. It's a great pleasure to talk to you today and give you a quick overview of this laboratory-based Master's Programme. If you're watching this as part of the UCD Open Day, then I will be available online to answer any questions. Otherwise, please do not hesitate to contact me using the email address that you can see on the screen. As well as teaching physiology on the medicine, science and BHLS programs, I have a particular interest in translational research and the application of molecular and cellular pathophysiological techniques to the study of diseases such as heart failure and lung fibrosis. I've been supervising PhD, MD and research masters in UCD for nearly 20 years and developed this taught MSc 3 years ago with a very specific goal of providing a program that offers in-depth one-on-one training in research skills and practical research techniques. This program is designed to offer a unique learning environment that will allow you to perform and perfect fundamental laboratory techniques and apply them to current research questions in an active world-class research environment. A particular strength of the program is that rather than running large group classes in a teaching laboratory, we offer one-on-one teaching under the supervision of a principal investigator within their research group. These PIs will often be in the lab with you themselves, or will have research teams made up of PhD students and postdocs that you will work with on a day-to-day basis. This will give you really excellent hands-on experience in the lab, but another benefit is that it can lead to very productive group tutorials when we come back together to discuss the ongoing lab work and the more theoretical aspects of the various techniques. For example, 4 different students in 4 different laboratories may all learn the same technique, but each lab will usually have its own tricks and tweaks to the protocol. So it's always interesting to come together and debate the pros and cons of the various refinements. It's a bit like setting you a baking challenge and asking you to go off and learn how to bake a cake with your grandparents or whoever the family star baker is. Each teacher will likely tell you that they have the best recipe in the world, but the chances are that when we bring all that knowledge together, we can all improve. And even if we can't improve the recipes, it's fun to judge the cakes and debate their merits. If I can stretch that analogy a little bit further, the emphasis during this program is on baking the perfect cake, not just churning out rubbish. In other words, we want you to take your time and perfect your laboratory techniques so that you can generate robust and reliable data. Whilst you'll be working in a research lab and pursuing real research questions, there is no essential requirement to generate novel data. In other words, success in this program is not about producing a substantial research thesis, like you would have to for an MSc by research, but becoming more confident that the data you do generate is robust and reproducible. With all the health challenges we face in the world, it is really important that we can trust our biomedical research data and our biomedical research scientists. For example, just look at what the COVID pandemic has taught us. It's a great example that highlights in a very acute way how important accurate PCR-based testing is at the moment, the importance of robust clinical trial data upon which we can define our vaccination strategies, and the need to keep up with COVID variants in order to develop new vaccines. Now, just imagine all the chronic health problems that affect us at the moment that need good biomedical scientists to apply their knowledge. So, is this Taught Masters for you? In short, if you're interested in a biomedical research-based career or a career that requires knowledge of how lab-based research works, then this is a great option. In particular, if you're interested in pursuing a PhD, this program offers a perfect foundation. In many ways, this program is very similar to stage 1 of most PhD programs. Usually, when you start a PhD, much of the first year is spent learning the techniques that you will apply to testing your PhD hypothesis in stage 2. This MSc will give you those skills and a good bit more. For example, it will give advanced training in areas such as scientific writing and communication. Also, if you decide to go on to do a PhD, you'll be able to take advantage of some transferable credits that you can use to get a head start on the taught component of most PhD programs. In fact, 75% of our graduates from the master's program have gone on to do PhDs, many of them within the School of Medicine. And it's been a real pleasure to keep in touch with them as they advance their research careers. Saying that, this program is not a commitment to an academic research career. In fact, one of the motivations behind developing the program was that I had heard from colleagues in places like Abbott and Charles River, and these were colleagues that had gone into industry after completing PhDs, that they were a little bit frustrated with the BSc graduates that were applying for jobs. and moving into the industry because they have relatively poor lab skills. Based on several related discussions, I thought there was a fantastic opportunity to provide a 1-year intensive practical research training period that would prepare students for a career in almost any biomedical arena and really set them up with a powerful advantage over BSc applicants. As I've already mentioned, a key feature of this program is that we offer one-on-one training in an active research environment. This graphic briefly summarizes the laboratory aspects of the program. When you first start in the program, you spend a couple of weeks getting to know us and reviewing the various laboratories that are offering positions. You then pick a topic or a laboratory that will provide the thread that links all your other modules together. The idea is that whilst you do not focus on your major applied projects until the 3rd trimester, you spend the first 2 trimesters learning the techniques and building up towards the main project. By spending most of your time working with one team with a particular research interest, you get a much more cohesive program. I'll go through a few of the modules in a moment, but in general, trimester 1 involves a little more time out of the lab with tutorials, workshops, and online teaching. But as you settle in, you start to do more lab-based modules and then are usually 100% in the lab by the summer trimester. In terms of the research topics, we have a large number of active researchers within the physiology team. This is not an exhaustive list, but we have several labs interested in respiratory diseases, such as pulmonary hypertension, emphysema, fibrosis, and COPD. Cardiovascular diseases, such as heart failure and hypertension. We have expertise in hemostasis and thrombosis and the role of platelets in diseases such as stroke and myocardial infarction. We have 2 very recent appointments with a particular interest in neurophysiology, learning, and dementia. We have several groups that focus on epithelial barrier function in the context of the gut and conditions such as inflammatory bowel disease. Extending from that, we have particular strength in the biology of oxygen and carbon dioxide sensing and the role of hypoxia in fibro-inflammatory diseases and cancer. So if you have a particular disease interest, it is likely that we can find you a matching supervisor. I will end with a quick overview of the different modules that will be either core components of the program or offered as options. Firstly, most of our research groups use cell-based models in their work. So you will learn the fundamentals of cell culture and how to grow cells that you will need for your research. You'll learn about primary cells, transformed or cancer-derived cells, and their application to the study of physiology and disease. Importantly, cell culture is a skill, a little bit like driving a car that you just need to practice. So while we'll cover the basic teaching in trimester 1, you will likely go on and use tissue culture throughout the year. Similarly, we'll cover the fundamentals of Western blotting, PCR, real-time qPCR, immunohistochemistry, immunocytochemistry, with an emphasis that suits the interests of you and the lab that you are based in. Each module then will be customized for your particular research interest. For example, you may develop a quantitative real-time PCR assay for a particular gene of interest, or learn how to use PCR to clone a gene and overexpress a gene in a particular cell type. Through learning and practicing these techniques, you will be required to work on assay optimization and experimental design. Those core laboratory-based skills then will be complemented by modules that focus on more fundamental research skills, such as scientific writing and communication, critical appraisal of the literature, online research skills, data analysis, and research ethics. A lot of these modules are spread over the year and use a complement of online teaching and face-to-face teaching. The final suite of modules that are available to you are designed to provide a strong foundation in various cutting-edge research techniques, such as flow cytometry, biological imaging, and proteomics. These modules are offered by world-leading experts who run the core technology platforms in the UCD Conway Institute, and you'll get to see and maybe play with some of the best equipment in the country. We will also keep you up to date with the latest research via the weekly Conway seminar series, extend your understanding of the importance of scientific integrity as you acquire your own data, and give you a flavor of how research discoveries can lead to the exciting world of innovation and entrepreneurship. Whilst the commercialization of research is not a primary aim of this program, it is absolutely clear that we all need to know the basics of how to develop and protect our intellectual property. Many of you may end up in industry or university spinouts, so spending a little time with our colleagues in the UCD Innovation Academy will give a great introduction to this world and may open some exciting opportunities for you. In summary, we think that this is a fantastic program for anybody that is interested in biomedical research. The emphasis is on practical lab-based teaching, supported by small group tutorials that develop your critical research skills and expose you to advanced research techniques. We are now looking to enroll students for a start in September 2021, and I'd welcome inquiries from anybody who has or is completing a BSc in a biomedical subject. If you've had some lab experience as part of your degree, it may help you get started, but research experience is not necessarily required. Many thanks for listening, and I look forward to chatting with some of you soon. Bye for now.